Everyone, it's Dr. Hall. Lecture 3 is all about action potentials. And in the first part, we're just going to set the stage so you can kind of understand the things at play to allow an action potential to happen. So this is our outline. For 3A, we're going to talk about setting the stage. So we're going to remind ourselves about the plasma membrane being semi-permeable. We're going to talk about ion channels, which allow ions, which are charged particles, to move across that plasma membrane. And then talk about the electrochemical gradient, which is going to determine the direction of movement of those ions, either from inside of the cell to outside or vice versa. And then something called the equilibrium potential. I'll leave you with a little bit of a cliffhanger there. So plasma membrane. It is semi-permeable. We have those that phospholipid bilayer, and so small and nonpolar molecules can cross freely, no problem, in and out. They have a pass to the club, they're on the list, it's totally fine. But if you are a charged molecule, like an ion, then you cannot cross without help. So that's where ion channels come in. So ion channels are these really big protein complexes that span the whole width of the plasma membrane. And they have a pore or an opening, think of it kind of like a tunnel through which the ions can travel. So it creates a little doorway, it creates an opening so that ions can move either into or out of the cell. Now, a couple of things about these. One is that they're passive, so it requires no energy then for the ions to move when we're talking about ion channels. They're also selective, and they can be open or closed under different circumstances. So I'm going to talk about those two things a bit more. So they are selective. An ion channel for chloride ion is only an ion channel for chloride ion or for sodium ion, right? So they are selective for a specific ion, and they use a couple of different features of different ions in order to exclude the ones that they don't want and only allow the ones that they do want. One is the size of the ion. So if you have a really small pore, if you have a really small opening, then only really small ions can travel through it. So that's going to help you discriminate. The other is electrical charge. So sodium and potassium are positively charged ions. Chloride is a negatively charged ion. So you can put different charges inside your ion channel that will repel or attract the charge of the ions that you're selecting for. So those are pretty straightforward. That kind of makes sense. But then you say to yourself, well, how could I make an ion channel that will allow large molecules in, but not allow small molecules in, right? So it's got to have a big pore size. So technically then small ions could come in too if they're the right charge. Well, there's this really interesting thing in chemistry called a hydration shell. So ions are charged, which means they're going to be hydrophilic, so they're going to interact with water. Now, so they're surrounded by water molecules, right? We're water-based organisms. Everything is in an aqueous or water-based solution, so there's tons of water molecules around. So anytime you have these ions, they are going to have water molecules that are around them and held around them due to these charges. Now, what's interesting is that in the smaller ions, like so we've got a sodium ion shown here, it is physically smaller, those bonds with those water molecules tend to be pretty tight, whereas potassium, which is larger, it tends to be kind of more diffuse and not as tight. So that the ability to separate the ion from its hydration shell allows some channels to allow in large ions and not small ions, which is really cool when you think about it. Okay, so we talked about how they can also open or close and what are the triggers that will cause them to open or close. So for the purposes of action potentials, we're going to talk about 3 different ways that they can open or close. Technically, there are 4 different ways that ion channels can open or close. I'm going to show you the 4th one here, which is mechanically gated. So mechanically gated ones, literally there's like something pushes on them. It's a mechanical or a physical stimulus that causes the channel to open. We're going to come back to those again when we learn about the senses. So a lot of our sensory receptors are going to have mechanically gated ion channels. For our purposes, talking about neurons, what we're going to be looking at is ligand-gated ion channels. And in the case of a ligand gated ion channel, that is a mouthful, a molecule has to bind to it. So in the case of neurons, that's usually going to be a neurotransmitter. So here you can see a neurotransmitter molecule binds to the channel, and so it opens. And when there is no neurotransmitter molecule bound to the ion channel, it is closed. So that's ligand gated. Another one is voltage gated. Now, I like this diagram because it's got all four in one place, but it's not really showing you that there's a change in voltage. It's not very clear. But voltage-gated ion channels will open or close in response to a change in the electrical gradient. So we've already talked about kind of the membrane potential, right? That difference in charge between the inside of a neuron and the outside of a neuron. So when that charge gradient changes, then voltage-gated channels will open, depending on what they're particularly set for. Then there are always open channels. And so they're often referred to as leak channels because they're just always open. So things are always leaking through. We'll see those as well when we talk about the movement of ions in neurons. So ligand gated, a molecule binds to them and they open. Voltage gated are going to open in response to a certain electrical potential. And then our leak channels are always open. So now that we have a channel through which ions can flow, we need to be able to understand which direction they're going to move. And what's going to determine that is something called the electrochemical gradient. So remember, second law of thermodynamics, entropy, nature, just natural laws of physics, right? Things are going to move out until they are as uniformly distributed as possible. So when you learned about diffusion in anatomy and physiology or human biology or other biology classes, right, a molecule is going to move from an area where it's highly concentrated down into an area where it's less concentrated until it all evens out. I think of it as kind of like Mother Nature wants everybody to be equal. So this is just a thing that always happens, is passive, right? And this is going to occur until the concentration is even on either both sides of the membrane or in all places of the solution or whatever it might be. Now, the same is actually true for electrically charged ions. So as you know, positive attracts negative, negative attracts positive. So ions are going to move until we have all the negatives paired up with positives, right? So the net charge is all going to be evenly distributed. So when an ion channel opens, ions are going to move according to those two gradients. A chemical gradient for how concentrated that particular molecule or ion is, as well as an electrical gradient looking at all of the charges, the positives and the negatives. So electrical gradient, remember the inside of a cell is negative 70 millivolts, so that is negative. Sodium ions are positive. So the electrical gradient of a neuron is going to make sodium ions want to move to the inside of the cell, right? That positively charged ion is going to be attracted to the negative charge inside the cell. What about the chemical gradient? Well, we need to know the concentrations of sodium inside and outside the cell. So it turns out that sodium concentration outside of the cell is 140 millimoles, which is actually quite high. And inside the cell, it's only 15 millimoles, which is really quite low. So it might look something like this. And so then, of course, sodium is going to want to move inside of the cell according to its chemical gradient as well. So in the case of sodium ions, both the electrical gradient and the chemical gradient will want to push sodium ions inside of the cell whenever ion channels open, okay? This is what that looks like in the diagram from the book. So we can see that here in the extracellular, outside of the cell space, we have lots and lots of these sodium ions. And on the inside of the cell, intracellular space, we have very few. So therefore, my chemical gradient is going to want to push sodium ions inside of the cell. We also know that the inside of the cell is negatively charged with a membrane potential of negative 70 millivolts, and sodium ions are positive, so that is also going to want to push sodium ions inside of the cell. Now, obviously, I drew these arrows just going through the membrane. They can't do that. They have to go through the ion channel, and if the ion channel is closed, they cannot move across. All right, so for sodium then, both the electrical and the chemical gradients are acting in the same direction. And so if those channels open, sodium ions rush inside of the cell because they have a lot of forces that are trying to push them inside the cell. Let's take a look at potassium because here the situation is a little bit different. So first of all, I went in a different order than I was expecting. Hold on. First of all, potassium is also a positively charged ion. So since the inside of the cell is negatively charged, that electrical gradient, as we can see here, is going to want to push potassium inside of the cell. So the electrical gradient will want to make potassium ions go inside of the cell. The positive is attracted to the negative. But if we look at our concentration gradient, here we go. It turns out that the potassium ion concentration outside of the cell is only 5 millimoles. That's super duper low. And inside of the cell, it's 150 millimoles. So it's 30 times higher inside. So that chemical gradient is going to push potassium out of the cell. So in this case, we have two opposing forces, right? The electrical gradient, negative attracts positive, wants to pull potassium into the cell, but then the concentration gradient, the chemical gradient, there's so much potassium inside of the cell, wants to push potassium outside of the cell to where it's less concentrated. So we're at loggerheads here. We're kind of fighting each other. We're having an arm wrestling contest, and the question now becomes, well, who's going to win? Which opposing force is going to be stronger and determine the net movement of our potassium ions? The way you answer this question is with math. So it's something called the Nernst equation, which allows us to solve for something called the equilibrium potential. Let's get into it. All right, this is all about helping us understand which way ions are going to move. So there's a lot of math here, right? There are some constants here. There is definitely, we're going to talk about the concentrations of the ion inside and the concentration of the ion outside, because that's going to let us know our chemical gradient. Then there are some other things that are calculated based on the permeability, as well as the temperature of most cells, which we usually set at physiologic temperature, right around 98 degrees Fahrenheit, 37 degrees Celsius, and gas constants. So there's no math prerequisite for this class. We're not going to actually do the math, but what I want you to know is that this mathematical formula allows you to figure out the relative strength of the influence of the concentration gradient and the electrical gradient based on the charge of the ion, based on all of those things. So you can figure out something called the equilibrium potential. So the equilibrium potential is the membrane potential, the cell charge level at which those two opposing forces are going to be balanced, right? So if you're having that arm wrestling contest, it's where you're actually like both winning equally. So it's going to tell you where those things balance out. So that will be the membrane potential at which the flow of the ion going in is matched by the flow of the ion going out. So some are going in, some are going out, but they're keeping in balance. So the ion is considered at equilibrium. The concentrations will stay the same. Let's take a look at that. So again, it's figuring out who's going to win. So remember potassium. So we had a high concentration inside the cell, low concentration out of the cell. So the chemical gradient was pushing out. And then potassium is positively charged. The inside of a cell is negatively charged when it's at its resting membrane potential. And so that was going to pull potassium ions into the cell. Who's going to win? All right. So we're going to use Nernst equation and solve for the equilibrium potential. So here's my neuron. Potassium concentration inside is 150, outside is 5, and I'm going to do 3 examples. So there they are. So in this first one, the concentration gradient is going to push potassium outside of the cell. But I'm going to set the membrane potential equal to the equilibrium potential. So the Nernst equation, if you solve for potassium, will tell you that it's negative 91 millivolts. So if I make the inside of my cell negative 91 millivolts compared to the outside, Nernst equation tells me that is the value at which I will have that negative charge pulling in just enough potassiums to equal the number of potassiums that are going out. So at negative 91, I have enough negative charge inside the cell to pull some of those potassiums into the cell, even though it's against their chemical concentration gradient. So at that equilibrium potential, there's no net movement. Every one that goes out is balanced by one that comes in, so the concentrations are going to stay the same. But it takes negative 91 millivolts to create enough of an electrical gradient to pull potassium back in to make that balance happen. So in this next example, we're going to look at your standard resting potential, your standard membrane potential, which is negative 70 millivolts. So negative 70 millivolts is less negative than negative 91. So it is less able to attract those potassium molecules, those potassium ions, into the inside of the cell. So negative 70 is less negative. Therefore, I'm going to have less electrical gradient pulling potassium into the cell. So now my little blue arrow is going to be smaller because that amount of movement coming into the cell is going to be less than the amount of movement of potassium going out of the cell. So therefore, the net movement of potassium is going to be out of the cell. More potassium is going to move out of the cell than into the cell. I hope that makes sense. All right, our third example. Concentration gradient, again, is going to want to push potassium out. But let's say that we have what's called a hyperpolarized neuron, like really hyperpolarized. Let's say it's negative 100, okay? So negative 100 millivolts. So this is even more negative than the equilibrium potential. And the more negative it is, the more able I'm going to be able to pull those positively charged potassium ions through my ion channel. So I already knew that negative 91 was my equilibrium where the two arrows were going to be equal. This one's even more negative, more powerful. So I'm going to have a bigger arrow, which means that if I had this membrane potential, I would have a net movement of potassium ions into the cell. So you can see, so remember we talked about how neurons are electroactive, right? Their charge is going to change. And depending on what that charge is, will determine, in addition to the concentration gradient, which way ions move. And the action potential is all about ions moving. So let's look at sodium. So sodium is kind of the simpler one. So we'll start with resting potential. So at baseline, the sodium concentration inside of a neuron is about 15 millimoles and outside is 140. So a very big concentration gradient that is going to push sodium ions into the cell. They're going to want to move into the cell if they're given the opportunity. Resting membrane potential for a neuron is negative 70. Now that's negative membrane potential and sodium is positively charged. So again, that's going to act to pull sodium into the cell. And so we know, given the opportunity, if the ion channel is open, then sodium is going to move into the neuron. Let's take a look at another example. So again, here's my neuron with my sodium concentrations inside and out, which will tend to pull sodium into the cell. And if we look at the equilibrium potential, it turns out it's plus 60. So using the Nernst equation, that's how you get that number. So you solve for sodium and you get its plus 60. So if I set my neuron at plus 60, which is like never actually happens in normal life, but if I could, then there would be no net movement of sodium because that positively charged inside of the cell would be enough to push as many sodium ions out according to the electrical gradient as many sodium ions were coming in according to the concentration or chemical gradient. So that would be no net movement of sodium ions into or out of the cell. All right, what about a negatively charged ion? So chloride is a negatively charged ion. Let's take a look. The equilibrium potential, according to the Nernst equation, for chloride ions is negative 66 millivolts. So here's my neuron. It turns out that the chloride concentration outside of the cell is about 120 millimoles, and inside of the cell is 10. So this is fairly similar to sodium in terms of like, it's almost like a 10-fold difference, much more concentrated outside the cell than inside the cell. Therefore, my chemical gradient is going to want to push chloride ions inside the cells to kind of equal out those concentrations. So equilibrium potential is -66. So what that means is at -66, there is enough negative charge to push those chloride ions out, because a negative charge is going to repel a negative charge. This is a negatively charged ion. So at -66 then, those two arrows are going to be equal in size. I will be pushing out as many chloride ions via the electrical gradient as I will be pulling in via the chemical gradient, and so I will have no net movement of chloride ions. So using that as a baseline then, I can figure out what's going to happen at a resting membrane potential of -70. So here's my cell again with my chemical gradient, which is going to push chloride ions into the cell should an ion channel for them open. If I'm at -70, so negative is going to repel a negative. -66, whereas I was equal. This is more negative, which means it's going to repel and push out the chloride ions even more. So I'm going to have a bigger arrow now. So my net movement is going to be out of the cell. So you can use equilibrium potential to figure out where those two forces are equal, and then based on whatever membrane potential you have at the time. So we keep using -70, but things are going to change when we start talking about action potentials. Then you can look and see, is this more negative than the equilibrium? Is this more positive than the equilibrium potential? And then what does that do to our electrical gradient? So using the equilibrium potential, we know that that is the membrane potential at which the ion is at equilibrium. The ins and the outs, the electrical gradient and the chemical gradient, are equally opposed. We're tying with our arm wrestling contest. So anytime the membrane potential, the VM, is different than the equilibrium potential, the ions are going to move. There's going to be net movement either into or out of. If it's a positive ion and the membrane potential is more negative than the equilibrium one, then you know that more of them are going to move into the cell. If it's a negative ion and the membrane potential is more positive, then that will move them more in. So you have to kind of play around with it. It's kind of fun. All right, so in summary, setting the stage, the plasma membrane is semi-permeable. You need channels for polar molecules and charged ions to move through. So we have these ion channels, which are made of these big proteins that span the membrane, and they're selective for both size and charge, right? So they can be very picky about which ions they let through. They can open either in response to a molecule binding to them, those are ligand-gated ion channels, in response to certain changes in the electrical potential, so those are voltage-gated, or they may always be open, those are the leak channels. Then we talked about the electrochemical gradient, which is going to determine where these ions are going to move when channels are open. The chemical or concentration gradient, the electrical gradient, right? So you've got positive or negative, and how does that relate to the charge of the inside of the cell? And keeping in mind, sometimes those work together, like sodium. Sometimes those work in opposite directions, like potassium. So that equilibrium potential is the membrane potential or charge inside of the neuron at which those two opposing forces are equal and there's no net movement of the ions. So we can use that value to predict which way an ion is going to move depending on what the membrane potential of the neuron is at that moment. Now, of course, then also given those ion concentrations for the inside and the outside of the cell. If those change, then you have to redo the Nernst equation and you're going to get different numbers. So it's a lot. Take your time, make sure you understand it, because then it's going to help you understand what's happening when we do action potentials. See you then.